This nerve connects with another nerve at that place we call a synapse. Synapses are junctions between two neurons and between neurons and receptor or effector cells, except that if that effector cell is a muscle, right, which we will talk about, I think, next here in class, right? If it's a muscle that this nerve is attaching to, we call that a neuromuscular junction, but it's no different, right? Here you've got the end of one neuron. What would that be? That would be the axon, right? And most likely a small branch of the axon, but so that's what this is. This is axon, right? What this is right here could be a dendrite, or it could be the nerve cell body of the other one. But all of the membranes of all of the parts of a neuron have these sodium and potassium gates and sodium potassium pumps that we've talked about. Okay, but now we got a few other players here at the axon. Okay, here remember we also have the distinction between a presynaptic neuron and a postsynaptic one. That word's not here, but so this neuron whose axon we're looking at here, this would be the presynaptic one, right? Because this area is the synapse, and this one is before, the synapse in terms of the direction of the impulse, and so this would be the postsynaptic neuron down here, okay? So what happens here, as you can see, is little vesicles that have these chemicals called neurotransmitters in them, because they are transmitting an impulse from one nerve to another, or maybe a muscle, right? Or maybe a gland cell, okay? So these vesicles are going to do the exocytosis thing. They're going to be moved right to the edge of this. What moves them? Cytoskeleton parts. We'll connect that too, right? What's that take? ATP. All this takes a whole lot of energy, right? To where they open up and they release their neurotransmitter into this little tiny gap that's called the synaptic cleft or gap. That synaptic cleft thing didn't make the syllabus here. They just said they release this neurotransmitter into the synapse. The synapse could be referred to as this whole setup here, like I drew the picture around, or if you want, like they're doing, they call the synapse just this space. Notice synapse means connection, but notice these are not touching, right? The nerve cell membrane of the presynaptic neuron is not touching the nerve cell membrane of the postsynaptic neuron, or if it was a muscle cell, right? So they have this space that the neurotransmitters are released into, and then this postsynaptic one has molecules that are the right shape for that to attach to. Here we have the receptor idea again. Just like a hormone kind of thing, and here's where it gets very confusing. Some hormones are also neurotransmitters. Adrenaline would be one of those. That's both released by a nerve cell to send this message, or released by a gland, the adrenal gland, to send the same kind of message, right? The message that is going to be triggered by that receptor signal molecule, right, attaching to some kind of correctly shaped receptor. That signal molecule, if I said receptor there, I meant that signal molecule, in this case neurotransmitter, attaching to the right shape. There's a couple specific ones they want you to know, but here's the simplest way that a neurotransmitter can transmit this nerve impulse from this neuron to this neuron. And that is when it connects to a sodium channel and opens the gate. So that's what this is. This shows the simplest kind of system. They can get more complicated, where the neurotransmitter directly causes the opening of sodium gates. What does that mean? It's getting messy here, right? But what that means is that enough sodium gates open here on the postsynaptic neuron's membrane and lets enough sodium in to get from resting potential to -55 boom, we are going to have an impulse in this postsynaptic neuron. Okay? This impulse has been sent from one nerve cell to another, or again from a nerve cell to a muscle. Here, while the picture is up, and I know it's getting messy, we got to throw this in as well, this part of the story. The nerve impulse you see in this white arrow, what's that mean again? It means sodiums and potassiums are going in and out, in and out, in and out. Impulse triggering the next part to trigger, the next part to trigger. Well, as it gets close to the end here, what this nerve impulse also triggers is calcium gates to open. Calcium becomes a player here that we haven't talked about. At the end of this here, there are calcium gates. And when the calcium gates open, like we're going to see with muscle cells in a minute, the calcium gates are actually what trigger the cytoskeleton to push these vesicles to the edge. So this is an important player in this game. I think it's going to come up in red ink in just a minute. Okay. The calcium part of this story happens right at the end of the presynaptic neuron, causes the movement of vesicles with neurotransmitter in it right to the edge to release those neurotransmitters and therefore make it possible for that postsynaptic neuron to fire. That story together with this. Here we have this idea now. This neuron that we're talking about, right there, that might be attached to by 10,000 other neurons. So it might be getting messages from 10,000 other neurons. But probably not all at once. It's probably getting maybe an impulse from 6,000 of them or whatever, right? Now we have a summation idea. It's getting impulses. What does that mean? Again, sodium gates are opening. But enough sodium gates have to open to get that little area that's receiving this impulse, right, to negative 55. So if it gets a few sodium gates open, but we get to negative 60 but not negative 65, that's not going to cause that impulse to really be an impulse. If after a little time goes by, we get another little bit of sodium gates opening, but it's still not enough, our nerve is still not fired. But look what happens now if the same impulses that by themselves didn't open up enough sodium gates, if they happen close enough in time to each other, temporal, they add up. So this one got it this far, and then this one came right on its heels, opened up enough more sodium gates to, boom, to do that, to get the threshold, to open up a whole bunch of sodium gates and happen that. Okay? What this is also integration. This one nerve cell, kind of like a tiny brain, is integrating a whole bunch of signals that might be coming to it from a whole bunch of different nerve cells, and it's not making decisions, but it's kind of like making decisions, and the decision is simple. If I get enough of these excitatory messages, impulses from neurotransmitters that open up enough of my sodium gates to get me to negative 55, boom, I'm going to do something. So now, here we represent two different neurons that I said could be 10,000 different neurons, right? Both of them are giving an excitatory message here. What's that mean? Both of them are releasing neurotransmitters that cause sodium gates to open, right? But if they're too far away, I'm going to start with this graph. If they're too far away from each other, then a few sodium gates opened. I'm sorry, forget what I just said. I'm going to come back to that one. So if they're too far away from each other, I'm going to put one of them here. Mike, I'm in the middle of doing a thing here. Sorry. And another one here. then we're not going to get enough sodium gates open in this place to cause a spike. But if they're closer together, spatially closer together now, then when that impulse comes at the same time from each one, now they're close enough to get me to negative 55, which shoots me up to there. And that's spatial summation. So once again, each nerve cell is like a little brain. It's got the ability to integrate all these different signals that if they come fast enough to the same place or to different places but they're close enough together to where they sum up to open enough sodium gates, boom, we'll have a spike. Now to this one. What if some of those nerves are sending a neurotransmitter that pushes you further away from negative 55? Well, now if this little nerve cell, like a brain, like we said, is getting some messages that say don't fire and some messages that say do fire, then it just depends on the amount of them. And the example they've drawn out here is that I got a fire message, but I got a not fire message, and that didn't put me close enough to negative 55 for me to fire. This is the kind of, again, integration that happens at each nerve cell. It's like what your brain is doing when it gets messages that say, don't do this, don't study, study, don't study, study. Which message is going to win? Well, I have to let you decide that one. So here we go back to acetylcholine, right? And we're going to talk about its secretion by exocytosis and then reabsorption at synapses. It's a very, very common one. It's also the neurotransmitter that is what's released by the nerve cells that connect to muscles. So at the neuromuscular junction, it is the signal molecule, but it's also one that's secreted between two nerves. And here you see an example of something that we're going to see over and over. The same molecule might cause exactly different kinds of responses depending on where it hits and what signal transduction pathway happens in the cell that it hits in. Okay, so here is that specific. I think somebody who wrote this syllabus did their doctoral dissertation on these things. Or they just picked it out of a hat as one of a whole bunch of different examples of this. And what this is that if a molecule is close enough to the same shape as another one, it can attach to the receptor, the same receptor as that other one, and in this case, it would not make something happen because it blocks that receptor from the real thing that actually triggers the receptor, and that's how this system works. So if we just look at the bottom half of this one, we see another aspect of how this system works. Once this acetylcholine is released by exocytosis, lands on enough receptors to open up enough sodium gates to hit the negative 55 to cause, again, this neuron to start doing action potentials all up and down it, right? You don't really want this message to keep being sent. And so what has evolved here in this case is there's an enzyme, acetylcholinesterase. There's an enzyme that breaks this down, and then the little pieces get reabsorbed and put back together and get reused so that this nerve can fire again and cause this nerve to fire again. That's the system. We're going to see some neat video of this also. But what this does, that they chose to put in the syllabus, these neonicotinoid, they're like nicotine, pesticides, right? What did nicotine evolve as? It evolved as a tobacco plant defense against insects, and this is exactly what it does. What nicotine does as well as these pesticides, right? They block this, and so therefore they keep sending the message. They keep sending the message. What do the insects do? They twitch, all their muscles twitch because they keep sending the message to twitch, 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 twitch. They don't get broken up by acetylcholinesterase because they're not close enough in shape to have it break it up.